I'm going to talk to you about how to do statistics. It's your, third day of being at DivSci, so this should be like exactly what you wanted in your lab. So the way that you start a statistics lecture, I've been to a lot of them, so I'm going to speak with authority here, is you show people the normal distribution. It has lots of lines on it. It's very exciting. Everyone's probably seen a normal distribution before, so I don't need to explain it. And then here's something that looks like a normal distribution, but it's actually a probability mass function. It's what you would use if you were doing discrete counts. And this particular one is for probability 13 Poisson distribution with a lambda of 10. I'm sure you all know what that means. And these are all of the different ways that you can do modeling. So if you are going to be doing a model, you need to choose what distribution you're going to use, if you're going to use a discrete distribution or a continuous distribution. And all of these can be interchanged in between each other. So to go from a Poisson distribution to a gamma distribution, all you have to do is change the conjugate priors. I don't need to explain that to you. You guys have clearly got this. So what we're going to talk about, obviously, is the negative binormal distribution. This is the equation for it. For our purposes, all we really care about is this. And obviously, I do not need to explain to you how we got from there to there. It's very clear. This is, I don't know if this is your experience of statistics in general, but this is my experience of basically every statistics lecture I've ever been to. There's some numbers, things happen, some things flash, they say, but all you really have to care about is this, and then I sort of like pass out. So that's what we're not going to do, because I don't like it when people pass out at my lectures. It makes me feel bad. So we're going to talk about statistics in a very different way. This is a famous quote from Ronald Fisher, who is an old dead white guy who you may or may not care about. But he said something kind of cool, which is, if you consult your statistician after your experiment, then most of the time, the only thing that that statistician can give you is why your experiment won't work. They can't fix it. Math isn't magic. This is a paraphrase. And so most of the time, you don't really need a statistician at the end of your experiment. If you set up your experiment correctly, then it's reasonably easy to analyze. You might need to learn some new distributions. You might need to go and learn some of that stuff from the first few slides. But the most important thing you can do for your statistical analysis is to set up your experiment in a way that it can be analyzed. So what we're going to actually talk about is experimental design and how you can make it so that your statistics doesn't hurt when you get to it. So let's say this is your postdoc. Doesn't he look exciting? So you are the PI of your lab. This is a thought experiment. Your postdoc comes to you and he says, I've got this idea. We want to do an experiment. We're going to have 144 individuals. Cool. We have 3 treatments, so we're going to put 48 in each treatment. Everybody's cool so far. Each of these treatments lasts a week. All right? We want to get this done pretty fast. So I looked. We have 3, and this is a choose your own adventure. If you work on bacteria, it's incubators, you work on plants, it's greenhouse space, whatever you work on, insert it here. You have 3 of those, okay? And each one of them holds 48 things, whatever the thing is you work on. You have 3 tanks, each one holds 48 fish, okay? So he comes to you, he says, all right, we've got these things, so here we go. I've designed this experiment. This is what we're going to do. In thing 1, we're going to put the red treatment. In thing 2, we're going to put all the green treatment. In thing 3, we're going to put the blue ones. It's going to be super simple because it will be really hard for me to mess up which treatment goes in which one, right? I'm always doing the blue treatment to these ones. It's going to be great. Reactions.
The control.
Red's control.
How do last time have they seen you voting?
So how do you feel about this experimental setup?
I mean,
Is your reaction, so I saw at least one of these. Well, what about the So, how would you fix this plan? So I want you to, this is an interactive talk. I don't know if I mentioned that at the beginning. I only give interactive talks. I would like you to turn to a physical person nearby to you and talk for, I don't know, 30 seconds about what you would say to this postdoc. How can you take his base experiment and make it not turn, talk. I am now going to solicit opinions from the press. So, somebody, just for the purposes of making this easy, somebody who wants to tell me how to fix this, just like wave at me and I will give you permission to shout. Yes?
You need to have each of your treatments in all three places so that you know that your results are just different environments.
Right. So we probably don't, so the thing that we don't like about this, the reason that someone immediately shouted bias at me, right, is if I do this experiment and this incubator, tank, greenhouse, place of your favorite organism holding area breaks, catches on fire, something like that, your experiment is what? It's not going to work, right? Because you've lost an entirety of one of your treatments. So we need to do something to split out our individuals across our places. We want to do something more like this, right? Did anyone come up with something different than that? I mean, you might have ordered your colors differently, but is that the basic solution everyone got? Right? So this is a fairly common thing that you, like, no matter what you've experimented on, you've probably set up something like this. And so it shouldn't have felt like a super difficult question. So we're going to make this experiment a little bit more complicated. Your postdoc here, he owns 3 undergrads. And they are going to be the people that are physically doing the data collection, whatever your data collection is. So, postdoc, as part of his plan, is planning to have undergrad 1 do place 1, undergrad 2 do place 2, and undergrad 3 do place 3. How do we feel about that? We don't like that. What are we going to do instead? So somebody give me an example. Yeah.
So each of the 3 undergrads does 1 color. So they have one of them doing the red, place 1, 2, 3.
Okay.
One green.
Yeah. So we could have somebody do this one, and this one, and then this one. I don't know how that one and that one, however it works, so that they're tiled. Okay. Other solutions?
Completely random.
You rotate them that student takes measurements from H1, then 2 days later takes
Okay, if we're doing timed, we could have them split which times they do. What if we're only doing one measurement? Talk amongst yourselves for a second. We're going to measure them once. I don't know why, because it's not a lot of All right, who came up with an answer? All right, somebody, who came up with the answer? Yes.
One undergrad for each color.
One undergrad for each color. Okay, so if we have one undergrad do all red ones.
Yeah.
Let's say I've got like 3 undergrads, right? One of them is just bad at stuff. I mean, it's not their fault. They're just not good at things. I didn't know that when I gave them the assignment, but after we got done with the experiment, I go and I look, and we were measuring these fish with this ruler, and the other students, they noticed that like the end of the ruler has some space where there's not lines, and so they moved the fish over a little bit, and this other student just didn't like the ruler thing, they didn't get it. So all of their measurements are just like not right. So that is that a problem? Yeah. So we don't want to have we don't want to give like student 1 all of these and student 2 all of these and student 3 all of these, because we don't necessarily know that all of our students are going to do exactly the same thing, and we don't necessarily want them to be able to mess up that much stuff. Yeah.
Our group had the idea to have all 3 undergrads do all of their measurements.
Okay.
And then you could average them, or just pick one.
Right. Okay. You could have everybody do everything. That's one good solution, because if everybody does everything, then you can average across. You can get rid of these human effects, right? When you're doing an experiment, you have the effects of your treatments, you have the effects of where you put stuff, you have the effects of the people you have handling it. The only downside to that is that you have to pay everyone to do everything, and so that's not super great. Yeah.
Don't we have an idea you could have everyone do a little bit, and then you could make sure that all of their values are similar enough for like mathematical analyses, and then once you have that confirmation, then you can kind of split up the work.
Sure. So you could do a trial. That means you'd have to set up the whole experiment twice. Right? So you'd have to do a little one and then a big one, which might work. Like if you're doing it like a whole bunch of them, then that's probably cheaper than having everyone do everything, right? Did you have a?
Yeah, so I have 2 thoughts. If we did have each one do a different place, then couldn't you lump their differences in with an overall block effect?
Maybe.
Or I would probably do it completely random.
Okay. Have them do it completely random. Did you have another thing? All right.
Maybe for this core experiment, you have different percentages. So maybe for one student to do one specific percentage and another one to do the second. And so every this algorithm it will do the same experiment, same procedure, then your data will be more different.
So just have one of them do all of it?
One or them to, for example, your experiment includes 3 procedure, and then the first one to do the first one, the second one.
Are you describing this?
Like, for example, if that cell culture and the treatment with medicine.
Oh, have each one of them do a separate part of it. I see. Okay, that's a solution too. I hadn't thought of that. My solution is this. So I have it set up like this, where I'm doing the same thing I did with my treatments, but I'm tiling over my students the other way, right? So I have each one of them measuring a third of each place. We came up with some other solutions. This isn't the only solution, but this is a solution that works really well for the rest of my math examples. And so this is the one we're going to use. So if you do something like this, the useful thing about this is that if you put all of your data into a big table, you get all of your samples. So I'm just showing you the first 9 rows. For each treatment, I have the red treatment, it's in place 1 and Riley measured it. The green treatment was in place 1 and Sarah. And I have this, each row has different information in it for all of the factors that could be affecting my experiment downstream. And then I have whatever my measurement is. So let's say that you were going to collect RNA from each of these things, whatever they are. What other variables would you record? So right now we've got treatment, place, and student. If we're going to do RNA, what else should we record? And for those of you that have never stood in a lab and done a DNA or an RNA prep, let me just show you what those steps are so that you can chat about this with everyone else. So if you're going to collect RNA, you take your plant, animal, microorganism of whatever description and squish it. So you have total RNA. You have to do a purification step, which usually involves running it through some beads. You have to sonicate the bits to fragment them or run them up and down through a string. There's several PCR steps to adapt, to put on adapters, and then a final PCR step to make sure that you have enough RNA that you can actually run it through a sequencer. So there's all these different steps. Most people, if you gave them this protocol, because I have to stand and physically squish my things, and then I have to put them in individual tubes with these balls, and then I have to sonicate them in a machine. Since there's these steps that are individual things, most people can prep at most 8 to 16 samples at a time. You've got 144. If you've got a fancy machine, then the most you can prep is a 96 well plate. So there's some variables and a picture. 30 seconds or so amongst yourselves. What else should we add to our data frame of variables that we should keep if we're going to be doing an RNA experiment? So we recorded which student did it. We recorded where we were housing each of our organisms. What other things should we do?
together. And
Okay, so what samples were each batch for our processing? Okay, what else?
So I would add RNA concentration
Okay,
and RNA impurity numbers.
okay, so some quality control numbers, yes. What else?
The state of the sample,
Okay,
pressure present.
yeah, how long this sample has existed, whether you froze it or not. Especially if that differs between your samples. Yeah,
the settings on all the different machines that you use.
okay, the settings on the machines, especially if you change them between samples. Did you have one back there?
Are you saying that all of the organisms are the same, or are these different organisms?
I don't know your life. If they're different organisms, that is good.
On that same note, like, I don't, there could be a system of each student is doing the exact same organism, like in the exact same order.
Okay, yeah, so that's a thing, like, are these 3 students splitting up these preps, right? Are they all equally good, and like what order they did them in and that sort of thing? What else?
The volume of your sample or how much you're using.
Okay, how much you're gonna put in the sequencer? Yes. How much you used before you switched it? Also good. I have a lot of technical terms. This is, I think you guys came up with most of these. So what your samples are, where you got them, how long it's been in storage. If you ended up using 2 different kits for prep, maybe that some of these ones were for this kit, and the other ones were from a new kit. How long it's been in storage, that sort of thing. Okay. Everybody, this all makes sense so far? All right, so I've done all of this. I'm gonna go put it into my sequencer. Is this how you load your sequencer? No. Excellent. I hit go too fast, but and this is for the exact same reason, right? Because if lane 2 fails, we're going to lose all of these samples. And the whole point of RNA-seq is to be able to compare our samples across compare how many reads we got from this one, how many reads we got from this one. If this lane doesn't fail, it just only gives me half as many reads as this lane, just for reasons, then I don't want to call all of the samples in this lane as having twice as much expression as these ones, right? So I need to track not just my experimental variables from my experiment, but from the thing that a lot of people think of as an assay. So you probably want to do something more like this, right? We could do the same thing we did when we were splitting up our samples across lanes or across our sample locations across our places, right? The thing that's cool about the sequencer is that the solution we had over here, that we could just have everyone do everything, is way more economical because the sequencer we're going to run once, right? And it isn't like our 3 undergrad. It doesn't take 3 times as long if we just have everybody run all 144. We'll get less sequence from each lane for each of these, but when we add them all back together, we'll get the same amount of sequence we would get if we had them split before that. Does that make sense to everybody? So if you're going to do an RNA-seq experiment, you would want to collect all of this kind of data that we had before, but also things like your sequencing date and the sequencing lane, and whether you pooled samples, which samples you pooled with each other, if they're run on 2 different sequencing centers, like which ones came from which center, and all that kind of stuff is important variables that you're going to want later, especially if one sequencing center that you hired is really good, and the other one had like an electrical storm during your run, right? So the important, yeah.
Right, quick question.
Yeah.
The 2 sequencing lanes, the 2 ways that you load it, would you always do the second option if you had the choice between the two? And have you seen a big difference when you?
So it depends. So I think Illumina right now can index. So if you're going to put all 144 samples in this lane, that means you need to be able to deconvolute them later, right? So that means that you have to have enough indices, enough barcodes to be able to do that. I think that the limit per lane is like 200 or so right now with Illumina. So it depends on how many samples you have. It's just, if you have 50 samples, then this gets rid of the variation between lanes, and getting rid of variation in your experiment is a desirable thing. So the thing I want you to take away from this little bit is that we want to think of RNA-seq as an experiment. It's just part of your overall experiment. It's not an assay. It's not like a thing that happens. It's got all of these variables inside of it that can change how your data is, or what results you get out of it. So let's say we've done this experiment. We have our matrix. It's got a lot of the same data that we had before, except now what we're looking at, I've simplified this a whole bunch. Pretend that for whatever reason, you got back all of your read data, you mapped it, you did all of your bioinformatics stuff. And what you have now is the read counts for just your favorite gene. So normally when you do RNA-seq, you process all of your genes simultaneously. We're gonna, for the purposes of the next 20 minutes or so, look at just one gene, okay? So that's what our read count is. So if I take the counts for that one gene and I plot them so that read counts is on the y-axis, and I just plot them by row number, I get a box, a scatter plot that looks about like this. It's got lots of dots in it, yeah? So now my experimental data, it's in this scatter plot and there's a line on it. What I want to know is how you feel about that line. Yeah? Yeah? So why do you feel that way about that line? Yeah?
It doesn't seem to have any correlation, purpose, whatever you want to call it.
Like, why is it there? Yeah. So, when we put a line through a scatter plot, what does the line mean?
The trend.
Okay, it's like the trend. What is it? What else?
It's a relationship between X and Y.
Okay, it means there's a relationship between X and Y. Is there anything else that you could describe the line as? The best fit? Okay, why is it the best fit line? This is not like a math question.
I mean, like, it depends on how you get the line, but it should reduce the error between the line and where the points are.
Yeah, okay. So if I put this line on here, this line's better, right? So the reason this line is better is because we have a best fit line and we're trying to reduce the amount of space between the dots and the line, right? So when you fit a best fit line, when you run a linear model, you make this best fit line, what it does is it goes and for every dot, so it draws a line. Then from every dot, it measures how far each dot is from that line in both directions, squares it so that it doesn't just cancel out, and tries to minimize that value so that the line is perfectly in the center of the dots as much as it can possibly do. What that means is that the line is the mean of your data. It's the average. So when we're doing linear modeling, we're drawing a line, we're drawing a line across here because it tells us what the mean of our data is. Does that make sense? That's why this one feels weird, because it's supposed to be telling us about the trend of our data, and it is clearly not. So a Bessel line in a scatter plot shows us the tendency of the dots. So that's where their middle is. And it's the average of all of those dots. Yes? So this is if I do it with a real LM instead of me just drawing a line in PowerPoint. And so I have the blue line, which is our mean, right? And then I have this gray stuff. What is the gray stuff? Yeah, it's like a confidence interval. I don't know how it's calculated offhand, but it's telling us about how much the line could move, right? So on this little dot, it could tip up and down that much in either direction, and still basically that's within the mean of our data. Yes? So this is if I took read count and modeled it by 1. That is the model we're using right now. If I model by treatment, that would be a useful thing to do, right? Because what we care about when we're doing an experiment is whether our treatment did something. Yes? So, if I model by treatment, what happens is, so I should point out, for those of you that actually know how linear models work, that this is not, strictly speaking, how linear models work. This is philosophically how they work, so that you can get a feel for what these different things mean. So what essentially is going to happen is we rearrange our scatter plot. So now the rows are in a different order. So now we've got all of our treatments grouped, and we get a new line for each one. So if I model read count by treatment, I get 3 means, which means I get 3 lines. So 3 best fit lines for all of the dots on my plot. Does that make sense? So mathematical modeling, at least with these categorical variables, so things like red, blue, green, is really just estimating the average for the data subsets that I tell you. What do you expect to get if we model treatment by student? So we check to see if our students are measuring differently. Talk amongst yourselves. 10 seconds. Go, go, go. What? How many, like, what do I expect to get out of All right. How many lines do I expect to be plotting now? How many lines? 9. So if I plot read treatment by student, I get 9 averages. Yes? So my question is, what is happening to my data here? Oh yeah, no, Riley's our problem child. I'm with you there. Yes, there's a problem child in this data set for illustration purposes. So as I'm adding terms to my model, as I am making more and more things in my model that I want to know averages for, what happens to my data? It breaks it up. Okay, we know more about where the variation is coming from. Right now we can point at and say, well, Riley is really a lot here. But for each one of my, for each thing that I'm modeling by, I model by smaller and smaller subsets of my data, right? So every time I add factors to my model that I want to model by, I am using smaller and smaller subsets of my data. In a real linear model, you don't actually lose them this fast, blah, blah, blah. But you're using less and less data to find these numbers. So your variation is also going to get bigger, because as you try to ask about the means and variance of smaller and smaller numbers, the variances inflate. Yes? So it is important when you are setting up your experiment that we try to minimize the number of things that we have to try to model out, right? Because we don't want to get to a point where we're adding so many things to our model at the end, at the statistics end, that what we're actually doing is asking for the average of like 2 points. Does that make sense? So it's important when you're thinking about how you're going to set up your experiment. If you want to set up your experiment to measure or to like look at treatment and what species it was and which lake you got it out of and 2 or 3 other things, if you've got 5 terms and you also have to add in that, oh yeah, we did it across like 11 lanes and also I had like all of these different students do it. Every time you add terms, you're making it harder and harder and harder to estimate the terms in your model. You're making it, your statistics much, much harder. Does that make sense? Excellent. There was another point I wanted to make about this. Oh, it was this. I was going to say that, well, this was up. I did. Okay. So let's say I also add in place. So what do we expect to happen there? You can just shout at me at this point. It's going to be a lot of them, right? So what we're asking for is all of these different means. It's going to end up where we're actually getting data about very, very small bits of our data set. Yes? So when you're setting up your experiment and the stats person that you talk to says, I would like you to do a power analysis, they're not trying to hurt you. What they're trying to get you to do is say, all right, if I'm gonna want to know about these 5 variables at the end. Just saying, oh, I'm gonna have 144 individuals, so that's probably enough, isn't helpful. If I have 144 individuals and I want to model by 3 things, I'm only really getting 5 or 6 individuals in any one of those. So power analysis is a way to tell you how big you have to make your experiment at the beginning, so that when you get to the end and you're parsing out all of these different things to find out where the variation you care about is, you can actually still do that math. Does that make sense? That's what I just said here. So it's figuring out how much data you need at the beginning that you're going to care about, so that when you get to the end, you can actually ask the question that you want to ask about. So I love this slide. You can talk amongst yourselves for a second if you need to, but why does this slide exist? What am I trying to get you to see here? Talk amongst yourselves for a second. I'm going to get a drink. All right, what does this picture show me? I only heard like that words were happening, but not what they were.
Two different models here.
There's, yeah,
We have two different models. We're trying to see the effect of lane or date is more influence on our data.
all right, sure, that's an interpretation. Which one is? Okay.
They're the same.
They're the same. They're the same. So if you follow every dot in here, they're exactly the same. So if we go back to our data frame here that I've been showing you, every time I had lane one was always run on May 23rd. Lane 2 was always run on June 30th. My variables are perfectly confounded. Sometimes statisticians say that to you, and they don't tell you what they're talking about. Those variables are perfectly confounded. And what that means in our, let's try to draw out what a linear model does in graphs, is that we're trying to make averages over exactly the same points. So I can't tell the model, do treatment plus lane plus place plus LP date, because lane and LP date would be averaging over exactly the same dots. And so it can't. That's just not how math works. If I made one of my samples that was in lane 1 have run on 6:30, it still wouldn't work because I've got it mostly confounded. You have to have your data, you have to be averaging over significantly different dots for the math to be able to go. I know I'm using the most technical terms here. Yes.
Are you saying that's a bad thing?
Yes.
Because, well, then you don't have to account for both of those variables.
Okay. If I don't care about both of those variables, then yes, then I don't have to try to get rid of both of those effects. If I change LP date to species, then that means I've confounded the thing I care about with a thing I absolutely don't care about. Does that make sense?
Yeah, but you could avoid that by running the species on different lanes.
Yes. Well, if you ran them on different lanes, then they would be confounded. So you can't get rid of the lane effect if it's confounded with the species effect. So if I make lane, if I just erase this and call it species, let's make it like a variable we might care about. Yes? Yeah. And I'm going to pop out and go back here. I think I actually have this in a So, if lane 2, if my green samples, like green is the thing I care about, lane is the thing I don't care about, right? If I run my model and tell it to do lane plus green here, it won't be able to model both.
Yeah, I get that. I'm just confused, like, if it's variables we don't care about, like lane and date, why does it matter if they're
So if there are things we don't care about, then it doesn't.
okay, that's what I wanted.
Yeah, sorry. I should change that example to make it a thing that we care about.
I have they might work.
Go.
What if your reagents on 6:30 were horrible? Oh, hello. Hi. What if your reagents on June 30th like didn't work? And then now all of a sudden everything for June 30th doesn't work, but you can't pull it out.
Yeah. So any sort of interaction-y things. And I do have an interaction example in here, but I don't think we're gonna get to it, but that's fine.
When things go wrong, we care about those.
When
variables.
things go wrong, we can care about them, but it's easier to think about as like a thing you do care about and a thing you don't care about. You don't want to confound. Two things you don't care about and you know you will never care about, no matter if the world ends, then those you can confound. In practice, you're gonna have to confound some things. Like, that's just how life is. So it's better to confound things you don't care about. But yeah, your variables are confounded if they're averaging over the same points. Now, we're into a couple of fun exercises in decision making. So you can talk about this yourselves for a second. This is the same data. Which of these models is the one that you should use if these are your 4 options? So we've got just modeling by treatment. Blue is clearly going to have a much, much higher average. We can model by treatment and students so that we can subtract out some of our student differences. We can do student and treatment and what place it was. Or we could do place and treatment. Apparently just place and treatment.
I think the most powerful model here will be only between treatments, but we need to consider people who's doing that as a date and a place as random effects of one effect.
Oh, yes. We're not going to get to random effects today, though. Sorry.
So I actually took one of your modeling class, and so I would say we would need to look at treatment and students, but I know after we have the data, we're not supposed to pick a model. You're supposed to model beforehand to not buy.
Okay. Yeah. So, yes, you should. So you should pick the experimental variables that you're going to put into your model beforehand. A lot of the time, you don't know necessarily which things in your experimental pipeline might have been weird that you're going to have to account for later.
Well, then based on what you said, then I guess we
So the one with the most variables is a good answer. Let's account for lots and lots and lots of things. But I also told you that I wanted you to record all of those things about your RNA. I wanted you to record all of those things about your experiment. I wanted you to have all of these variables that you actually cared about. And I told you that what is going to happen to your data if you try to actually account for all of those? It is not going to work, right? So we have to pick and choose. So generally speaking, you want to pick a model that is going to have the fewest parameters that you can have while still answering your question. So just having a model that has every single variable that you can imagine in it isn't good. Having a model that has just one is often not good. There's a happy middle ground that you want to hit. In this one, I would probably pick maybe here. But that's because I cheated and already know that the other variables don't have very much effect. Good job.
So you mentioned that we want to record all kinds of. So you mentioned that we want to record all kinds of information when we're doing this experiment, like the settings on all the machines. Let's say we have 100 different knobs with 100 different settings. Is there a way to look at each individual variable or groups of variables to say this one is important or this one is not?
Yes.
Without biasing.
No. So you want to, so there's two things there. If you've got a machine with 100 knobs, you want to record where the knobs were. But if you haven't moved them throughout your experiment, then you're not going to include that in your model anyway, right? Because that's assay, that's thing that happened. If you're constantly moving them and you have 100 knobs, then you basically made your experiment so complicated that your sample size would have to be like near infinite, and you're not data science today anyway. I'm not sure.
Is there a way, so let's say there's something in between, maybe the centrifuge has 10 different settings, and for whatever reason, the centrifuge was changed. And so, it is what it is, you have to include that in your model. If you want to say, like, okay, maybe the centrifuge setting, in theory, shouldn't have a big effect. How can you tell if it does?
So the way that I do it is basically do like exploratory data analysis, which is more or less what I'm showing you here. I've just drawn lines on it, right? If you plot your rows by centrifuge settings, and they look like this, where like your centrifuge settings that go with Riley are like clearly causing some big effect, then you can add it to your model. But that's yes. Question?
Yeah, so in your model, whatever student you use, blue is always higher than green and red, and whatever student you use, green and red are probably not that different from each other. So if you just go by, if you ignore the student, then you have a huge variation. If you compare each student, then the variation is smaller, but you're breaking up your samples into 3 discrete sets, but you have 3 times a person who found that blue is always higher than green and red. What is more powerful? I mean, is there, which way should you decide to go? Does that make sense? In a way, having 3 students, each individual accounting who are always coming out on top, is that more powerful than having combined 3 students showing that all the data points of blue are higher than all the data points, although now the variation within blue is so much higher.
Okay, I see. Yeah. The way I have it set up, so that could happen in real data where one of your students is just causing a lot more variation than the others. In the way I have it set up, they all have exactly the same amount of variation. I just added 20 to these values. So it wouldn't actually change anything at all about this data. But if it's a variation problem, the real way that you would fix this is you just run it through a mixed model and you subtract out the variation from your students. Yeah, that's outside the scope of this, like, let's try to figure out what statistics words mean lecture. Yeah.
So it's kind of the reason for recording all these different variables is to be able to know what could possibly be confounding your experiment, right? So you identify which lanes you were running your RNA test in, you were recording your dates, you recorded who did what, so that when you do plot it against these different variables, you do see trends, you have a chance then to normalize your data,
yes,
right? And that's something that you didn't say anything about. Before you average is to normalize the data against these different variables, right? And also to recognize things that might be outliers. You might have one data point that's just way off that you know that can't possibly be good, right? And if you throw it in, and if you add it into your analysis, it's going to throw your whole experiment out.
right. So you can.
There has to be. You have to do more than just kind of accept it for what it is. You don't say, well, this is what we did, this is what we got, take it or leave it, right?
You can design it better from the start.
Right.
Right. But that's what I'm saying is that we are trying to design a very good experiment here by recording all the variables that could possibly be complicating the experiment, right? But now that we have that information, we should try to make use of it in order to fix the data, right? The readout is one thing, right? But the readout is skewed by certain, it could be place, it could be by person, it could be something, but the fact that we did record all this metadata, right, allows us then to go back and make corrections that'll make the experiment more valid. Does that make sense?
I think so. So that's effectively what the model is.
Basically, I'm having trouble with us being asked the question, which model should we use? Like, well, what question are you trying to ask? And also, it's like, I haven't done anything yet to control against these different factors. So we're looking at treatment versus student, treatment versus location, treatment versus color, right? And in principle, all the students should be equally good at doing things. So it shouldn't matter by student. It shouldn't matter by color, right? Because we're talking about one treatment, one measurement, right?
Okay.
So in principle, every single measurement should be the same for that one subject, right? No matter who does it, no matter where you do it, right? There should be one right answer.
Okay, yes.
Does that make sense?
Yes, but the way that you get rid of the variation, right? At least if you're going to be doing linear model, like the way that you get rid of the variation is by accounting for those metadata in your model. So asking which metadata are worth including is how you get your normalized data. Because like RNA-seq data, you don't generally, like there's no normalization that you do before you get raw counts. You put it into your modeling framework, and then you have to build a model to account for all of the variation that you don't want to be included into your counts. So being able to think of, like what I want you to think about here isn't like objectively in life is the red, like should we include student? I mean, it's, if you have this data, what sorts of things do you use to decide what model is going to work for your data, right? So if you have, up here I have place, sorry, and 1, 2, and 3, they look basically the same, right? I wrote this data so that they would look the same, but place is clearly not a thing that did affect this data, whereas student did. And you can have a different experiment where the opposite is true, like your students could all be great. Yes.
It's important for us to look at that and see if there's not a plan. Choose to setting a second.
Yeah, so it's just like, yeah, so I just wanted you to sort of look at some data and think about like what kinds of things would I consider if I was doing this model. Yeah.
Is there any mathematical model to decide, or mathematical equation to decide which value I should include in my model or not?
Yes, but I wasn't going to cover model choosing today. So if you have like I can answer lots of questions like this, but like the pictures is we were doing I was aiming for more of a like what do these terms mean? How do these things generally work? Or a framework of how do these stuff? Yes.
So 2 things.
Yes.
One is we need to.
I know. I was looking for my end slide. I was just going to click to it, but then.
The other is that Raina will be talking on Friday about model, to some extent, model choice.
Cool. Then I'm gonna